I want to start off by welcoming Dr. Andy Penn to give his talk about GRIN2B variants and synaptic dysfunction, the context of GRIN2A subunit expression in neuronal development. Thank you so much for being here.
Okay, so I want to thank the organisers very much for inviting me to speak in this session. So normally I'm at the end of the day, so it's refreshing to be at the beginning of the researcher day. So I'm the support act for the research sessions today. So Mara's already given you the title of my talk, and what this is really representing is an update on the sort of research that we've been doing looking at GRIN2B variants. And this project is funded by CureGRIN, and they are providing essentially their funds are being used to subsidize a PhD student who's dedicated to working on this project. Oh, skipped one. Okay, so I'm only going to give you two slides of sort of basic introduction because of the audience that we're speaking to today. So the context is the GRIN2B variants. That is what this talk is going to be about. And these patients have intellectual disability, hypertonia. They're usually non-verbal. Some have seizures, autism spectrum disorder, and motor disability. But essentially, these are neurodevelopmental disorders. GRIN2B is one of the one of the genes that encodes an NMDA receptor subunit, specifically one of the regulatory subunits of the NMDA receptor. And it's not that these suddenly patients, children have started getting this disorder. It's that we've only just started recognizing that their disorder reflects actually variants in GRIN2B. And so, in fact, in the NMDA receptors in general, we've been seeing, and actually a lot of other genes that we now know are involved in rare disorders. We've seen an increase in the discovery of genetic variants in the NMDA receptors over the years, and it's mostly thanks to next generation sequencing technologies and their development and economic application. So GRIN2B encodes one of the regulatory NMDA receptor subunits, which it is the glutamate binding subunit, and it assembles with the obligatory GLUN1 subunit, which binds coagonists, in particular D-serine or glycine, and their binding essentially activates the channel. And if the membrane potential is permissive and instead it's depolarized, then we can also get unblocking of the pore by magnesium and therefore activation of the channel and the passage of charge and charged ions through the channel. Now, there are those regulatory subunits actually can tune the NMDA receptor properties. In particular, I'm going to focus a lot more on the deactivation kinetics of the receptor. So I'm just showing two NMDA receptor subunit types here, GLUN1 with 2A in a diheteromer, and a diheteromer of GLUN1 with 2B. And you can see that these receptors with the 2B can stay open a lot longer than the receptors with the GLUN2A. Now, these two subunits that I've just illustrated actually are highly expressed in pyramidal neurons, so the principal neurons in the cortex and the hippocampus. But that's in the adult case, and actually during development we see, and we've known for a while now, that the NMDA receptor subunits can vary in their expression levels. And so early on in development you have predominantly just the GLUN2B subunit in these cells, and then during development there is a switch to include GLUN2A in NMDA receptors. And in fact, I'm picturing here 2 copies of GLUN2B or GLUN2A in the receptor, but you can also get 1 copy of each of those regulatory subunits. And something that Casper Hansen showed, who's one of our collaborators now, is that the receptors that have both subunits, GLUN2A and 2B, actually adopt sort of the activation kinetics of the receptor that are more like that of the GLUN2A only receptor. So this, we found this interesting. We thought, well, how does, if you think about genetic variants of the GLUN2B receptor, that if GLUN2A has this dominant effect over the NMDA receptor, could that essentially lead to differences in the function of the receptor when they've got the variant? So some work that my previous PhD student did, Marwa Al-Masri, she essentially did some experiments where she knocked out knocked out both GLUN2A and 2B, and then expressed different GLUN2B variants. So here she's got wild type GLUN2B expressed in the double knockout background, or R540H or R696H. And you can see that those 2 particular variants, they are prolonging the decay time course of synaptic currents in the CA1 of hippocampus. But if we only knocked out the GLUN2B and then replaced the GLUN2B with the human variants, then what we see is something very different. We actually see a bit of acceleration of their decay kinetics. So GLUN2A is dramatically changing the way that these receptors are, these synaptic receptors are responding. So it's not just, there we are. It's not just in neurons as well. Collaborator Casper Hansen was able to specifically express dihetramers or trihetramers. So dihetramers, GLUN1 and GLUN2A, or trihetramers, GLUN1, GLUN2A, and GLUN2B, in HEC cells which don't normally express NMDA receptors, then applied agonists to the membrane of those HEC cells, and sees that the activation is slower for these gain of function variants, just like we saw in the neurons. But when it comes to expressing GLUN2A as well to make these trihetramers, then in the case where we find that the deactivation time constant is a lot shorter, almost as short as the deactivation time constant of receptors that only have GLUN2A. So this got us thinking, okay, so if the properties, that properties of the gain of function variant, since GLUN2A is changing during development, could the way that the synaptic receptors respond differ at different stages of development? We've not technically done this. We've only so far looked at sort of knocking out or allowing the inclusion of the native GLUN2A. We don't really know to what extent the changes in GLUN2A is sufficient to drive these sort of polarized effects on the variant receptors. And also, the implications of this are that maybe at different stages of development, you might want to try different strategies for trying to correct for those variants. So this is, and the point I'm trying to make here is that this is meant to be the same variant, right? It's just at different stages of development. So we wanted to sort of see if there's any sort of, whether there's any sort of traction in this idea. And that is really the goal of this project, is to see whether or not it provides some proof of concept at sort of a fundamental science level, to see whether variants can actually, A, behave differently at different stages of development in neurons, and B, whether they can respond differently to different sort of drugs which are of interest to this community. So those experiments that we were doing were actually all in organotypic hippocampal slices. So we had been taking the floxed animals. I'm going to show here the floxed animal for GRIN2A, prepared hippocampal slice cultures, and then transfected individual neurons by single-cell electroporation with GRI, and in those other experiments, we were also co-expressing the GRIN2B variants. And then we would do whole cell patch clamp. Now, when we do this, we noticed that in some sort of preliminary work, we had looked at some different, for knocking out the GLUN2A subunit, the GRIN2A subunit, we had noticed, we had done some experiments looking at recordings at different time points after sort of in essentially what are postnatal days plus the days in vitro. And what we noticed was that we don't really see much change, much in the way of changes in the decay time constant of those NMDA receptor-mediated EPSCs. So this suggests to us that we've kind of already passed that developmental switch in this particular system, and so it's not really it's not in its current form at least suitable for us to sort of address these questions about development. So what we've done is we switched to using primary cultured neurons. These are usually prepared from younger animals. We can't prepare slice cultures really from younger animals without sort of sacrificing the effect that has on disorganizing it, on the culture. Those tissue becomes disorganized very easily when you get them from animals that are too young. So we've switched to using dissociated culture. Still in mice, we'd be taking them either at sort of embryonic or early postnatal, sort of day 0 or day 1 after birth, and prepare cultures from them and let them grow in vitro, and then patch clamp and record from them and record their NMDA-mediated spontaneous synaptic currents. And when we do that, we are able to see that change in the decay time constant of those synaptic currents. This is sort of mainly the fast component of the synaptic currents. It's quite difficult to measure the slow component in these sort of in spontaneous NMDA EPSCs. They're a bit smaller. And so we felt that this was a good sign. We are seeing sort of that the changes we might expect from a switch from a slow deactivating GLUN2B subunit to a fast deactivating GLUN2A subunit. And we don't at least see any changes in sort of the cell time constant of the voltage clamp, which might otherwise explain some changes in kinetics of the responses. So this was promising, but what we wanted to check was that indeed that GLUN2A is responsible in these cultures for that switch. So what we've adopted is the use of CRISPR. We're kind of moving away from using sort of dedicated mouse lines for using our genetic manipulations. So we took this tool from Rick Huganier. This is a dual guide Cas9 expressing construct, which has guides that target sequences around the intron, well, in the introns around the exon that has the start methionine for the native mouse GRIN2B. And expression of this in cells, usually co-expressing with another plasmid that lights the cell up green for us. We can then see that those cells should have the exon excised, and then NHEJ repair mechanisms should essentially rejoin those introns without that exon, and so knock out the GRIN2A gene. And we've done that in this experiment with these cultures, and we do see that there is a longer decaying time constant of the EPSCs after knocking out GRIN2A. So using the same sort of assays, we feel a bit more confident that what we're seeing is actually due to the NMDA receptor subunit switch. Now, one of the reasons why we've been moving away from the sort of the approach using the GRI GFP and overexpressing the GRIN2B variants is that we can't really properly control the levels of the GRIN2B that well, and we are concerned that, especially when you're looking at small changes in GRIN2A expression, we want to be maintaining sort of normal expression levels of GRIN2A. So what we've done is we've used the same CRISPR construct, but this time we are designing a donor that has so once it splices out the exon, we replace it with the human the open reading frame of the human GRIN2B, and then have an iris and GFP and a polyadenylation signal, so that we can then essentially replace that exon with this sequence, which generates an mRNA which can express both the GRIN2B variant and the GFP, all from the endogenous promoter. So we first checked this with the construct which Rick Huguenier had used this T-kit system to introduce set tag, and we checked that we can also see the set tag included in the receptors, and indeed it works for a few of the a good proportion of the cells that are transfected, co-transfected with M-cherry, and indeed we've now literally only in the last sort of month we've been able to test this construct, and this donor sequence we suspect is actually incorporated nicely into a good fraction of the cells. I think we had 60% of the cells that were both GFP positive and M-cherry positive. So these are knock-in of GLUN2B, human GLUN2B. And ultimately, we want to be able to do this for lots of variants. So the nice thing is that this construct, this donor construct, we can easily chop out a large fraction of the GRIN2B sequence and replace it with the GRIN2B sequence of a bunch of variants that have already been made in mammalian expression vectors and used by in emery. And we have just made the first one, so V618G, which is a poor mutant which relieves magnesium block, and it should do so of diheteroameric and triheteroameric NMDA receptors. So it would be a nice sort of functional, a good way to check for functional incorporation of the receptor and how quickly we get that. So that work is still ongoing. What we've been looking at with this functional dominance of the GLUN2A was for gain of function variants, and we've also seen that when we express loss of function variants in backgrounds without GLUN2A and with GLUN2A, we see some rescue of the synaptic NMDA receptor current in those loss of function variant conditions when there's GLUN2A. And so it did make us wonder whether there's a sort of a molecular chaperone effect of GLUN2A, whereby triheteromers with the GLUN2A may actually help deliver GLUN2B variants to the surface. And Casper Hansen's lab, I've got one more data slide to show, and his PhD student Lauren, they made a beta-lactamase tagged GLUN2B and have done an assay in some hex cells to sort of to see whether there's any sort of support for this idea. And so with the wild type, you've got with these absorbance measured over time of the nitrocephin that's been added to cells that express these constructs. This is the case without GLUN2A. We have total and surface, and so a good proportion of the receptors are at the surface when it's wild type GLUN2B. And here you see that the surface receptors are really low level for this loss of function variant. I'm skipping a bit there. But GLUN2A doesn't seem to really actually have much effect on it. And we've been able to see this for a few variants so far. And we've got some ideas looking forward that maybe there may be some other things in particular going on in the neurons. And we've got some other ideas of some other variants to look at here, which would be interesting to contrast with this. So just summarizing, we can recapitulate GLUN2A dependent speeding of NMDA receptor mediated EPSCs in cultured neurons. I've developed a scalable CRISPR approach to knock in GRIN2B variants in primary neurons. Work is ongoing to refine this and apply it to developing neurons, and our evidence in hex cells does not support a molecular chaperone role for GLUN2A. And here are my acknowledgements. Big thanks to Scott Kinghorn, who is the PhD student funded on this, makes it all possible. Thank you.
We'll do Q&A after.
Yeah, perfect. Yeah.
Thank you so much. We really appreciate your wonderful presentation. And just to reiterate, we'll do Q&A with them at the end of our three speakers here. Next, I want to welcome Dr. Maria Lania, who's studying the role of GLU A3 or GRI-3 AMPAR subunits in hearing and hearing loss with sex differences. So please welcome Dr. Maria.
Thank you, everybody. It's a pleasure really to be here and also to be able to share what we have been learning about the role of GRI-3. So in my lab, we are interested in how structure and molecular components affect function. And I'm going to be also talking about no central nervous systems. I'm going to be talking about hearing loss, it's not deafness. And this hearing loss mainly affects in aging. So I'm going to be talking about a very exciting period of time that is adolescence. So I'm going to try to do it the best of I can. Okay. So these slides just represent sounds very complex, but I'm going to try to summarize the best. So this represents the beginning of the auditory pathway. So to be able to hear well sound, so we have any change in the environment, right? It's going to move from or translate it from mechanical to electrical thanks to this sensory epithelia that is the inner ear cells in the cochlea. So then that signal will be changed to electrical, and it will be pretty much transport to the brainstem. And again, it's one single nerve, is the auditory nerve here in the ganglia of the cochlea, and then it's really transferred to this beautiful organized structure in the brainstem, the cochlear nucleus, and from there on it will move to the cortex. So again, this synapse, that is the inner ear cells on the auditory nerve, right, has to be very precise and to be able to encode sounds correctly. And for that, we need the AMPA receptors, right? So in this field, for a long time, we really didn't know exactly what is the potential role of the AMPA receptors in here, in addition that they need to be activated to be able to send information correctly. So the type of hearing loss that I'm going to be talking does not affect any sensory epithelia, right? That would be a completely deafness. It mainly affects the synapses. And a loss of synapse here at the base of these inner ear cells, it will go affect how sound encoding is affected through the whole central nervous system. And that will affect cognition, right? Again, from here on, it's a complex cross pathway that will reach the cortex. And I'm going to be mainly concentrated at this level at the very early of the beginning. So this just represents these inner ear cells, and this is a very basic structure of these afferent dendrites of the sparaganglion neurons. And mainly the AMPA receptor types are the 2, 3, and 4. So in adult synapses, the GLU A1 is not present there. So again, everybody knows in this audience that dependence of the GLU A2 editing might change the calcium permeability and sodium permeability. And what we know is in the cochlea, the GLU A2 edited is expressed, but there's also some indications that potentially homomeric receptors. But again, it's still speculations. So this is how it looks like at the electron microscope. It's a ribbon synapse. It's very different to the brain. It looks like to the retina. So we have a presynaptic ribbon with the anchor of synaptic vesicles and then a postsynaptic density. So this postsynaptic density that appears here in green is the one that contains the 2, 3, and 4 AMPA receptors. So many people have used immunofluorescence, and they have pretty much counted synapses in hearing loss. I'm going to be talking not only synapse loss, synapse loss, but also changes in AMPA receptors in the absence also of one subunit. Again, we need AMPA receptors to hear, and also people have shown that after noise exposure, right, excitotoxicity, AMPA receptors are involved. But again, the question is, what is the subunit that could play a major role? So when I started these studies, I was using rat, and we were using mainly in the brainstem to try to understand plasticity of conductive hearing loss. And then we will start seeing that the GLU A3 AMPA receptors starting being upregulated. Okay, and the first question is, how do we detect that? So then I was in Japan at that time, and I got the mice from Kenji Sakimura over there, and I brought them to Pittsburgh, and then started analyzing the hearing. And the way that we analyze hearing is just a neurological test. It's what we call auditory brain responses that is not invasive, but that allows to understand hearing thresholds against by intensity, and also how is the firing of the neurons along the auditory pathway. So for example, this first wave 1 represents the responses of the auditory nerve, following by cochlear nucleus, brainstem, et cetera, et cetera, et cetera. So again, by putting different intensities, we know about the thresholds. It's the same that you go to an audiometrist, or they put you different sound intensities and say, well, you hear or you don't hear, right? But at the same time, by analyzing against the amplitude of each of the wave, we know how the responses are of the firing in this particular location. Again, a hidden hearing loss is characterized by normal thresholds and a decrease in wave 1 amplitude. And what you see in here, when we compare, this in particular case are first the male mice, so that compare the GLUA3 with the GLUA4 KO, that particular only while both have pretty much similar thresholds to wild type, only the GRI3 knockout mice, so have a decreased wave 1 and wave 2 amplitude, right? So the GLUA4, pretty much nothing, right? So in that way. So this is starting thinking about, okay, so is the GRI3, is a fast AMPA receptor subunit that affects the periphery. So the brainstem is totally fine, but again, we don't know if it's a response because the first organ, right, it's already decreased. So this is around 2-3 months, so they are sexually mature. The question is, we knock down totally a gene, so we are not talking about mutation. I would really love to be able to understand how a particular mutation of the GRI3 affects that. So the question is affect development. So we tested this mice much earlier, right? So in 5 weeks old, something happened here. Well, 5 weeks old, the mice hear completely well. So we did a study to be able to understand potentially a mouse that is pre-symptomatic. And with that, we'd be able to understand if there's something that can lead to this synaptopathy in much older mice, right? And then I will not, because of timing, I will not go into detail, but very much what we show is that it affects mainly the maintenance of the synapses. So, but these are the males, so the question is about the females. So in the female, we did pretty much the same. The only thing that you can see here is that the hearing of this decrease in wave 1 amplitude occurs much earlier, right? When the mice are already in 5 weeks old mice. And it's pretty much maintained, right? Normal threshold relatively, but very low. And these are in relatively ambient in a facility that's quite noisy at the beginning. So now we compare these 5 weeks old mice. Now in magenta, you have the female. In dashed lines are the KO. And in blue, you have the male. So you can see very nicely how these females at these 5 weeks, they already have this hidden hearing loss. So the question is why this may happen, right? So we knocked out a gene, and another important thing to understand is how the other subunits, right, that they may contribute to that. So again, in the wild type, we have 3 subunits, but in the KO, we know that only there are 2. GLUA1 we tested is not present. So the way that they form the dimers and the tetramers is much more limited in the KO, right? So the question is, what is the contribution that affects synapses? So the way to do that, and that was in collaboration with Mark Rutherford and Washington University St. Louis, so we sent in the cochleas, and he did immunostaining for a presynaptic marker for the ribbon, CTBP2, and also for 2 and 4. We know that the 3 is not there. And then we can do synapse count and also expression intensity. And just by your eyes, you can already see how the expression levels is very different between the KOs and the wild types. This is in particular a male, but it's similar to a female. So this is a complex slide, but I'm just going to focus on here. We compare females and males. And what we see here is the amount of synapses, unpaired synapses, only with the ribbon, indicating already a disruption of the synapse is higher in the KO, but is both in female and male, but it's much higher in the female. Similarly, with synapses that they don't have ribbon, only postsynaptic density. And when we check about receptor subunits, only the ones that GLUA4 in theory are homomeric and potentially more calcium permeable. Again, they are higher in both the KO in the males and the females, but much higher in the females. So that means that synapses are starting being disintegrated, particularly higher and stronger in the female than in the males. In terms of intensity levels, so the lack of 3, again, affects the stereochemistry, the other subunit composition, not only in the number, but also in the intensity of those ones that they already exist at the synapse. And you see that there's a decrease of 2 and then the increase in 4, and that leads to a change in the ratio between 4 and 2. So there's potentially more 4 in those synapses, indicating more potentially calcium permeable. And that could lead to excitotoxicity. And just to show you that the female mice have what we call the swellings that are characteristic of this excitotoxic. So this is a normal looking synapse at the electron microscope. And when you see these swellings, those are abnormal synapses, that those are related potentially with excess of calcium and sodium. So the question is, why the females are more vulnerable? So far I have not been able to tell you, we still don't know why. So one thing, potentially, we know that the GRI3 is an X chromosome link gene, so a potential incomplete inactivation is possibility. So we know that in human, the fibroblast is incomplete inactivation, we know that. And then we know that the RNA, the GLUA3, is higher in the female at the sparaganglioneurons, but it doesn't really explain that potentially. The other things that there could be is that, it could be the sound levels. So again, this facility were very highly noise. So the question is if we decrease that ambient sound, if we are able to reverse the phenotype. And the other could be potentially sex hormones. So to do the first thing, what we did is to raise the mice, some of them in ambient after the postnatal day 20, in the same noisy facility, and another we keep them in quiet. And this is just to summarize, and this is just to let you know that at P35, so what you see here, the wave 1 amplitude, again, and the orange represents the KO in the ambient, and here in magenta is the GLUA3 female in the quiet. And you can see that in maintaining in the quiet, we reverse the wave 1 amplitude much, and what was very nice. And structurally, there are structural differences, but they don't have the swellings. And they also have increase in the GLUA4, GLUA2 radio, but it's much stronger. That changes in the ambient than in the quiet. So from here, we can say that this hidden hearing loss of synaptopathy is augmented by ambient sound levels, and the cochlear dysfunction in ambient sounds is prevented by rearing the mice in the quiet. The other thing was the sex hormones. So for that, we started doing, again, gonadectomies around postnatal day 22, so much, much earlier than the peak of the sex hormones. And then we maintained these mice in the ambient, and we did the kiss auditory brain responses, and also we collected blood. And this is unpublished, so I'm looking forward also to hear information about that. So these are the slides of the female versus the male. Let's start with the males. As you can see here, hearing thresholds don't change, but also doesn't seem to be an effect of the gonadectomy. The gonadectomy are the ones that have the white circles in between. So as expected, the wave 1 amplitude decreasing in much more older mice, not in younger, but again, there's not effect of the gonadectomy at all. However, in the females, where we see, again, these mice hear very well at P20, but when we do the gonadectomies, so these are the shams GLUA3 KO mice, and these are the gonadectomized GLUA3 KO mice compared to wild-type mice. As you see here, we reverse also the wave 1 amplitude, and seems to be also maintained in aging. So this suggests that there's a potential effect of estradiol on AMPA receptors at the inner hair cells, and in particular, there's increase in the vulnerability, and also the estradiol increases the vulnerability of the female GLUA3 KO to ambient sounds. And that is, we know that the sex hormones contribute to potentation of AMPA receptors in the hippocampus, And we know that can able also to increase calcium permeable AMPA receptors. So in here we have a machine that. And this is just to show you that we're interested about the hormone levels. We know that in the males from a group from Rodolf in Germany, that there's not much difference between the wild type and the chaos. We are going to confirm that. But the female, what we found is that the sex hormone levels are totally off in shams GLUA3 KO mice. So for example, estradiol is gone as expected after gonadectomy, but these are the levels in black of the wild types, and you see how the higher levels of estradiol, that are very abnormal levels. We're repeating this much more and also considering the estro cycle, but also that leads another potential role of GLUA3 potentially with ovulation. I know that GRI1 has been involved on that. I've been trying to get information about the GRI3, I didn't find it. There's some expression in the hypothalamus of GRI3, not a lot, but there's there, but that would be something very interesting to pursue. So this is just to summarize. So in here, I've been trying to tell you that the lack of a gene affects differently between the female and males. It affects synapse formation, and sometimes the defects that we detect, they will lead to hearing loss. And particularly that will potentially be more effective if, particularly in the females. And that is something that I'm really looking forward to learn more, but we know that the ambient sounds contribute to that, and also the estradiol also seems to be contributing to the increased vulnerability of this GRI3 female KO. And just to finalize, I'd like to thank many people past and present in the lab, and also collaborator Mark Rutherford, and the funding, NIDCT, and also the Pennsylvania Alliance Healing Foundation. Thank you.
Wonderful, thank you so much. So now we're going to have our final speaker for this first session here, Dr. Ludavica Montonucci, who's going to talk about predicting variant pathogenicity and functional effects in glutamate receptors, transfer learning from NMDA to AMPA receptors. Thanks.
So good morning, everyone. My name is Ludavica Montonucci, and I work in the LAL group in Houston. And the focus of our research is genetic interpretation. Does it go? Does it go? Okay. So I don't have to advocate to this audience why genetic interpretation is important. Variant interpretation is critical for designing. Sorry, can I move this? I cannot see my slides. You know where I am. So variant interpretation is critical for correct diagnosis, and also in order to target personalized therapies. And the two fundamental questions we want to know for variants are, of course, whether a variant is pathogenic, whether it can cause the disease, and if so, what is the molecular change that the variant is going to have effect on. There are still big challenges in variant pathogenicity, of course, and in particular, most of the variants have still no clinical significance, and experimental and clinical evidence is very slow to accumulate. So our approach in our group is to use computational methods, to train computational methods based also on machine learning in order to fill this gap, while clinical and experimental evidence is building up. So during today's presentation, I will present some recent and ongoing works that have been applied this computational approach to GRI research. So I'll start by presenting a predictor that we already built. I will describe some community engagement which produced the Green Portal. Then I will describe the challenge that we are actually tackling, which is prediction in GRIA genes, and then our approach to solve this issue through transfer learning. Sorry. Okay. So last year we published a paper in which we presented two machine learning methods that were specifically designed for variants in green genes. The first one is about pathogenicity predictor, and the second one is a predictor, computational predictor for gain or loss of function, increase or decrease functionality. And we found that despite we have many computational predictors that are general, like alpha missense or Revel, our computational predictor, the one that is tailored specifically to green gene, outperforms the existing ones. And also we presented the first computational predictor that is able to predict whether a variant has the likely effect of giving an increase gain or a decrease loss of function. And this is the first one for green genes. So this can give the possibility of knowing the functional effect without in absence of experimental evidence. So, and this was, we were able to do this just because we have enough data to train machine learning. And this data was aggregated in the Green Portal, which is a community effort that include clinicians, scientists, and families that allow to aggregate functional, phenotypic, and genetic data on a single resource, which you can access at this website. So in the Green Portal, you can have information about the phenotypic overview of your variant of the gene, of your variant or a specific gene or the whole green genes. You can explore the location where the variant is located on the structure. You can classify your variant, explore its functional effect from experimental data. And of course, there's also educational content, and you can download also a report for a specific variant of interest. So thanks to all these aggregated data, we were able to produce the two machine learning that I introduced before. And this is the main design of these predictors. So we have a data set of variant. We have two data sets of variant. One classified as pathogenic or benign, and one classified here as with increase or decrease function. We encoded this variant through evolutionary and biophysical properties, and what our predictor has that was not available in other general predictor is that we included some features that are specific of an MDA receptors. That is the distance of our variant from the ligand site, so glutamate glycine site or the magnesium block, and also distance from the membrane or from the pore axis. So we decided to introduce this feature because we observed that the population and patient variants distribute unevenly on the protein structure, with population variant clustering on the N-terminal domain, while patient variants cluster either on the ligand binding domain near the ligands or near the closure of the gate, near the M3 closure. And interestingly, the same happens for the function, in which variants that tend to decrease protein function happens in the ligand binding domain, while variants that increase function, that produce a gain of function, happen to be located near the gate, so in the transmembrane domain. So this is the design that we used. So our challenge now is that can we build a similar predictor for variants in the GRI genes. So we first studied and downloaded the GRI variant dataset that we have in public databases. So variant from Nomad, which represent variant from general healthy, supposedly healthy population. But we put a cutoff on allele frequency of 10⁻⁵, and we end up with a limited number of available variants. While in ClinVar, these are most variants, as you can see, are classified as VUS, and we have a really small variant which are classified. So 64% of GRI variants in ClinVar are of uncertain clinical significance. And of course, these are also a small fraction of all the possible variants that one can receive from a genetic test. So this data set, once we clean this data set by applying some quality filtering on allele frequency, removing duplicates, conflicting classification from clean variant nomad, we end up with this number of variants, which is insufficient for developing dedicated methods as we did for the green genes. So how can we do? Our idea was, can we use what we trained in the green genes and apply it to GRIA genes? And this is called, there are several methods called transfer learning in which these things are, this can be achieved. So transfer learning is a machine learning approach in which a model that has been pre-trained on a task is applied to a different task, provided the task is related. So we have a task 1, task 2 for which we have a lot of data, and we are able to train a machine learning, and the task 1 for which machine learning is not possible due to scarce data. So what we do is to use the knowledge of the problem with big number of data to adapt it to solve the first problem. And this is a strategy. There's no specific way to do it because it can be done in many different ways, by optimizing threshold, by pre-training, by re-encoding the feature. It can be done in several ways. However, this transfer learning has been used successfully in several fields, in several biomolecular fields, and this is the case, for example, for the chemokine, GCPR receptors, in which the high similarity between these type 4 and type 5 receptors allow to use what has been known on the expression levels of one protein to the other protein. So for type 4, the expression level were known for many variants, but they were not for type 5. So to transfer learning, they were able to predict the expression level in the type 5 protein, and this is the experimental and predicted expression level which show high correlation. So are prediction of variants in NMDA and AMPA receptors enough related to be able to make this transfer learning? We know they have the protein are very similar in structure, they have the same subunit composition, and so our aim is to try to make this transfer learning. And in particular, when we investigate how variants, population and patient variant distribute on the structure, we find the same distribution that we found for an NMDA receptor. So, however, we have to quantify this similarity. So we applied, we made a pairwise sequence alignment between all GRIA genes among themselves to see they have greater than 80% sequence similarity, and GRIA genes with green genes. And the sequence similarity greater than 40% is very high. And it gives us a one-to-one correspondence between residues in the GRIA and in the corresponding green gene. So our approach has been, when we have a new variant, for example, in a GRIA gene, first we find the corresponding residue in the green gene, and then we map those on the structure. And we retrieve the prediction for the corresponding green gene. And in this way, we have a pathogenicity predictor for the GRIA variants. So these are the results that we obtained when we applied this transfer learning for the pathogenicity predictor. So we applied two different transfer learning, one in which we retrieve the prediction when the residue is the same residue at the same position, and it's the same alternative residue is changed. So if we have exactly the same variant in the green data set, and sometimes the residue is different, so it's not possible to have the same alternative residue because if it's a single nucleotide variant. So we take the prediction even when just basically taking the same position. And we did this for global, local, and mixed sequence alignment. So the alignment type doesn't change, so we have these two blocks in which this transfer learning performs remarkably well, given that there's no other information. However, And it's incredible that it performs very well, the one in which the alternative amino acid is not taken into account. So meaning that what really drives the prediction of pathogenicity is likely to be just for these receptors, the position on the structure. Because in this second model, it's not even required that the original residue is the same, that the reference residue is the same. And so we achieve a very high AUC, which is a measurement of how the predictor performs from 0 to 1, and it's around 0.92. However, of 280 variants, if you see the last column is the number of variants that we were able to classify, so that were included in the alignment and for which there was a prediction in the green genes. And as you can see, we predict only a small number, so 99 or 195, which correspond to 34 or 68%. So our predictors still have a low, quite not so high coverage. So the next idea would be then to move to structural alignment, in which we can decide other rules to map, to build a one-to-one mapping from residue in the AMPA and in the NMDA. As you can see, when we aligned green 2A with green GRI2, we see that the Rotmy square deviation is very small, 6 angstrom, almost comparable, basically the double of the resolution of many structure, and they align quite well. So in the future, in order to increase the coverage of our predictor, we will move to make the mapping through a structural and not a sequence-based approach. So to conclude, so we successfully applied. So first, we developed two machine learning methods for green variants, demonstrating that having dedicated methods for specific genes increase accuracy. Second, that GRI variant interpretation remains a major unmet need, with most of variants with uncertain clinical significance. And our first project, by applying transfer learning for pathogenicity predictor, proved successful. It still can improve, but it lays good ground foundation to move to build the first functional predictor for GRI variants. So now what we are working on at the moment is expand the variant coverage using structural alignment, develop the GRI-specific molecular function, and finally probably adding another refinement layer in which other AMPA-specific and that are not green-specific features can be included. So yes, I would like to thank all the LAL group and all our collaborators, and thank you for your attention.
I'll invite all of our speakers up to do a quick Q&A. We'll do 10 minutes here. Yes.
I have a question for Andrew. It's a very nice talk. For the variant knock-in strategy, I'm wondering what the efficiency, because you're doing this in primary cells, so what's the frequency of getting the knock-in of the variants?
Yeah, so we did, we just co-transferred with the co-transfection marker, and actually we're trying to improve that by putting the sort of an M-cherry on the SB-Cas9 sort of as a fusion to make those numbers better. But despite that that donor being about 4 kb, which is about 4 times larger than what was being used before, we were still getting pretty good knock-in efficiencies of around 40%. Yeah.
Okay, thank you.
Yeah.
Other questions? Okay.
I have a question for Ms. Rubio. You talk about hearing loss in the mice with the GRI3 and also sex difference in that. Do you also see that in patients? Do you know? Because I tried to find, but I could not.
This is something I would be very interested. So we were not expecting to have such a strong phenotype. In particular, because for a long time, we didn't really know that the GLUA3 was present at these synapses. So it was just recently that we start finding that really plays an important role. So I don't know if because potentially this is the whole gene that affects much more, but I mean, I would be very interested to really address that with more questions. So this is not a deafness. So this is something that, for example, that you go to a very noisy environment and you feel that you don't hear correctly, like an aging, right? But this occurred much earlier, right? And I don't think that these are questions that pretty much are addressed. So when I discussed this with Megan Arnolds about the questionnaire to do, is that, well, it would be important to ask in noisy versus in quiet environments, right? And again, it's something that would be really interesting to, and I know to really know. And also, not many people really pay attention to the periphery as much. So that's why coming here would be great, because it's one way to understand much more, and also to potentially bring awareness that also there are sensory systems that they have on parareceptor. Yeah, but I would love to do. Yeah, thank you.
Question for Ludavica. How accurate is NOMAD these days in terms of making sure there's no patients in amongst all the general population? I heard that maybe its accuracy is somewhat on the wane. And so, for example, if we get contacted by a patient and there's a case in NOMAD, how many cases would you like to see in NOMAD before you could potentially say non-pathogenic without doing an experiment?
Yes, exactly. So that's why we put some hard filter on 10⁻⁵ allele frequency. But in NOMAD, there were in GRIA variants, there were like 2,000 variants, and we end up with 200 putting our filter. So we excluded most of them. So I do think there are some, unfortunately, patient variant in NOMAD, and so it's really needed to do a hard filtering. So we would have had, I think, around 2,000 variants at the beginning we started with. We end up with 200, so yes, definitely.
And just to follow up as well, so your numbers are getting very close to 1 in terms of your, the accuracy. Is there any, when you're going from like 0.91 or 0.92, have you still got room to improve, do you think? Is it going to be possible to make the predictions as nearer to 100% accurate?
So if we have if in theory it's possible, if the model have all the information needed. So alpha missense is already quite accurate. However, all predictors predict well the easy things and give no answer for the difficult parts. That's why we need to expand the coverage, because maybe we are doing very well because we are predicting the part of the protein that we know well how they work. There are other parts that probably we don't know yet. So this is the big challenge now, to be able to give a prediction for all.
And then one final one. Is there any possibility of including patient symptoms in amongst the predictions as well? In terms of if you know for a fact that a patient had a gain of function, could you use those symptoms to help to refine further?
Definitely, yes. This has been an approach that has been already used by the LAL group for the INSCN variants. It's definitely possible to use a phenotype as a proxy for molecular function. They have, I think, the paper on, yes, it's been used. We didn't do it yet now, because of course we have to have a big number to do that of data. But definitely, yes.
Yeah, I have a couple quick questions for Andy. Did I understand correctly that in the slices you didn't see the speeding with the developmental switch that you would have expected? Is that what?
Yeah, we didn't, but we only look to give the time for the transfection of and knockout of the GRIN2A. We were looking at between day 15 and day 30 or something like that. So that's day 15 and day 30 in vitro, and these were plated from mice that were at 7 days old. So
Oh, right, because I
So-
just couldn't remember the developmental time points for Camilla Bologna's paper years ago with Roger Nickel, where they have slices and they have they can drive the switch basically in young slices.
Right.
And I was just curious, is yours contradicting that, or it's just a different
No,
time point?
I think just the way when we're doing the manipulations and when we're recording, it's just a bit too far down the line to be able to look at that. Yeah.
Okay, thanks. And Lanya, I had a quick question. The GRI3 knockout is really fascinating, and I know that with more and more sequencing, the schizophrenia population is now implicated, GRI3 and GLUN2A, and they tend to be, as I understand it, null, like so haploinsufficient. So the ones that are schizophrenia associated, if I have that correct. Do you know of any of these hearing potential phenotypes in schizophrenia patients?
I've been asking that particular question to the people in psychiatry, like Robert Sweet, he does that. And then there's something there. There's some papers, but not really very clear information about that. So I would really like to know more. They don't do hearing tests and anything like that. So that's the thing. But because they have other issues that potentially affect how the hearing. But that is something that it should be sure, it should be done. Yeah, agree.
Any final questions? I'll ask a quick one. I'm just wondering about the transfer learning and if you think this could be applied for GRIC as well, or if it's too early.
In theory, of course, yes. I haven't checked how much data we have now, but of course, this is a great opportunity. So transfer learning, I think, is very useful exactly in these cases when we have tasks that are really similar because all the glutamate receptors are very similar. And so definitely, yes, it can be done.
Great.
The limitation is the function data for validation.
Right.
So because pathogenicity is not anymore the big problem, it's more like the function. And I think there's not enough function data that we can validate the models yet.
If everyone wants to give a round of applause for our speakers, thank you guys so much.